In this video, I will review DNA replication. So even though bacterial cells and eukaryotic cells are very different and organize their DNA in a different way, they generally use the same strategies to get the process done. The first thing to consider is that bacterial genomes are organized in a single circular chromosome. Remember, a chromosome is going to be a double-stranded DNA molecule, and it's really a single unit of DNA within a genome. Now, eukaryotic cells, on the other hand, will have many linear DNA molecules or chromosomes. So we can, imagine that there could be lots and lots of chromosomes within this nucleus. And importantly, they have ends to them, right? So all of these linear chromosomes have ends, circular chromosomes do not. So we'll see at the very end that eukaryotic cells have evolved a way to protect the ends of linear chromosomes during replication to prevent any loss of genetic information. So let's use the bacterial genome or E. coli genome as a model for how replication works. The first stage is initiation. There are a couple things that have to happen during initiation. First, the double-stranded DNA has to be unwound and opened to create a replication bubble. That happens at a specific sequence called the origin of replication. There is a single origin of replication in the E. coli genome. Again, once that origin of replication has been recognized, the double-stranded DNA is unwound, the replication bubble has to be stabilized, and there are a lot of proteins that help to do that. Second, primers need to be synthesized to create a free 3' hydroxyl. That's really the starting place for DNA polymerase. Okay, so this is what it would look like once a replication bubble has been stabilized. So again, this is happening at a specific sequence, the origin of replication. There has to be the helicase, which is going to unwind the double-stranded DNA. So of course, the helicase would first start at the origin, and then it's going to be acting at the two forks. So these are the forks on either side of the bubble. The bubble itself is this open part. Okay, so another enzyme called topoisomerase is going to be acting ahead of the forks to relieve any strain that is put into the DNA strand when helicase unwinds the DNA. Another protein that's needed is single-stranded binding protein. So this protein will bind to single-stranded DNA to prevent it from re-annealing to itself. So it's keeping it from forming those complementary base pairs and closing up the bubble. So again, that's essentially the first part of initiation. Then primase is going to come along and add complementary RNA. And that RNA is going to be made in the 5' to 3' direction. So for both of the leading strands, the little RNA primer is going to be made going towards the fork, and that's going to be on opposite sides. Then on the lagging strand, the RNA primer has to be going the other way so that it's going in the 5' to 3' direction. So initially there are 4 RNA primers that are going to be used by the DNA polymerase in order to synthesize DNA. Okay, so now if we just focus on one side, one fork, and we won't even look at the whole molecule, just at one little portion of it, we'll see how the directionality of synthesis leads to having a continuous leading strand and discontinuous lagging strand. Okay, so again, we're looking at just one fork of the replication bubble. Now, if we were to carry on this strand all the way through, it would go all the way through and there'd be another fork and then come back around in a circle. So this fork, the strand is going to be running 3' to 5'. And so the 5' end is here. And then this other strand would have to be running 5' to 3' since it's anti-parallel. That means when the RNA primer was synthesized by primase, it had to be going in this direction, 5' to 3'. So remember, primase can't put a primer in this direction because that would be not anti-parallel, and that's not how these molecules are synthesized. So instead, when primase put the primer down here to start, it had to go in this direction. So that 5' end is kind of hanging out there, and then the 3' end is going to be here. So both of these 3' ends have a free 3' hydroxyl that DNA polymerase III is going to use during elongation. So then the enzyme DNA polymerase III is going to hop on and it's going to synthesize complementary DNA in this direction on the leading strand. So again, this is leading strand synthesis. And on the lagging strand, it's going to synthesize in the 5' to 3' direction away from the fork. Now, remember that these aren't free ends here in this DNA, right? They're actually in the context of that circular DNA, the circular chromosome. So this would be carrying on to the other fork like that. And on this side, down here, there's going to be an RNA primer that would have been put down here, going towards the fork on the other side. So when DNA polymerase III is synthesizing discontinuous strand, cha-cha-cha-cha-cha, it's going to run into that RNA primer and then fall off. So at this point, helicase needs to be functioning at the fork in order to unzip all of this DNA that has to be replicated. So let's see what that looks like. Okay, so if Helix unzipped some more DNA, double-strand DNA, and it revealed this new template, well, on the leading strand, it's just going to continue on, right? So the leading strand is going to continuously make complementary sequence. But now we've got an issue because the lagging strand doesn't have anywhere to go, right? It would have fallen off here and it's got to replicate this. It needs a free 3' hydroxyl to do that. So primase is going to synthesize a new primer, and then the polymerase is going to hop on there and chug along this way until it hits that primer, and then it's going to fall off. So this is going to keep happening until all of the DNA has been replicated. Now again, if this is E. coli or a bacterial genome, it's circular and we're not going to have to worry about this end here because it's going to be continuous with the other side. But when we get to linear chromosomes, this is a problem, and we'll talk about how cells deal with that. So here I'm going to talk about how the DNA has to be processed before it's really been completely replicated. So the first thing that has to happen is these RNA primers have to be removed from the new strands and replaced with DNA. So RNA polymerase I is going to come along and it's going to chew back the RNA primers like this. And what that's going to reveal is a new 5' end and 3' end. DNA polymerase I can use the free 3' hydroxyl there and fill in the gap with DNA. Now, the thing it cannot do is form that last little bond to the 5' phosphate. So there's still going to be a little bit of a nick in the backbone in that area. So the same thing will happen with the RNA primers from the other strand. So all the RNA primers are removed, and the same enzyme will be adding them, adding DNA to fill in the gaps like that. Then the enzyme ligase is going to come along and seal the nicks in the backbone. So now there are two complete DNA molecules that have continuous polymers formed throughout. Okay, so the same first step in processing has to happen in eukaryotic during DNA replication in eukaryotes as well. So a DNA polymerase is going to come along and chew back the RNA primers, just like we saw in the bacteria. And then that polymerase will be able to extend from the 3' ends until it hits an end. So in this case, it's going to go in this direction and this direction there. All right, so then we've got some nicks that have to be sealed up, and that's going to be done by a ligase. So that's now two continuous new strands. But the problem is, at the lagging strand end, there's going to be some RNA primer removed, but there's no free 3' hydroxyl to actually fill that in. And so this is where the enzyme telomerase adds extra sequence to these ends to protect any genetic information. So again, this enzyme, you don't have to know how this happens, but it's going to use sort of an RNA guide, which is kind of weird, in order to extend those lengths. And then the extra stuff that's there will get chewed back. And now you have these new 3' and 5' ends of the DNA molecules. And now we've got a completely replicated and processed DNA molecule.